What if I told you there's a natural secret weapon against cavities, and it might already be in your own body? Today, we're diving deep into some fascinating new science that could completely change how we think about dental health. We're not talking about some new, complex chemical, but a simple, natural amino acid called arginine. So, how do cavities form in the first place? It's a story most of us know. We eat or drink something sugary, and the bacteria in our mouths have a feast. As they break down those sugars, they produce acids. These acids are the real villains. They slowly eat away at our tooth enamel, creating tiny holes that eventually become cavities. These bacteria don't just float around randomly. They're master architects, building dense, sticky communities on our teeth called dental biofilms. You probably know this better as plaque. Within these plaque fortresses, the acid production goes into overdrive, concentrating the damage right where it hurts the most, on your tooth's surface. For years, our main strategy has been to fight back with fluoride and by brushing away plaque. But what if we could make the plaque itself less harmful? What if we could turn it from a tooth destroyer into a tooth protector? That's where arginine comes in. Arginine is an amino acid, one of the fundamental building blocks of protein. It's naturally found in our saliva and in many foods we eat, like red meat, nuts, and dairy. What's so special about it? Well, certain beneficial bacteria in our mouths have a secret power. They use a system called the arginine deaminase system, or ADS for short. This system allows them to convert arginine into alkaline compounds. Remember how the bad bacteria produce acid? Alkaline compounds are the exact opposite. They neutralize the acid, raising the pH of your mouth and creating a less hostile environment for your teeth. When there's more arginine available, these good, protective bacteria thrive, while the acid-loving, cavity-causing bacteria struggle. It's like bringing reinforcements for the good guys in the battle happening on your teeth every single day. Early lab studies were promising. They showed that arginine could change the entire makeup of dental plaque, making it less dangerous. But lab studies are one thing. What happens inside a real, living human mouth? To find out, a research team at Aarhus University in Denmark, led by postdoctoral fellow Yumi C. Del Rey and Professor Sebastian Schläfer, designed a clever clinical study. They recruited 12 participants who all had active tooth decay. This is important. They were studying people who were already susceptible to cavities. Each participant was fitted with a specially designed denture. This wasn't for chewing, it was a mobile science lab. These dentures allowed the researchers to grow and collect intact dental plaque in a controlled way, right inside the participants' mouths. Here's how the experiment worked. The participants would dip their dentures into a sugar solution for 5 minutes, simulating a sugary snack. Immediately after, they would dip the dentures into one of 2 other solutions for 30 minutes. One was a simple distilled water placebo, and the other was an arginine solution. To make the results even more reliable, one side of the mouth always got the arginine treatment, and the other side always got the placebo. They repeated this routine 3 times a day for 4 days, allowing a mature plaque biofilm to form. Professor Schläfer explained the goal was to see how arginine treatment affected the plaque's acidity, the types of bacteria living in it, and even the sugary matrix that holds the plaque together. After 4 days, the dentures were removed for a close-up analysis. So, what did they find? The results were pretty amazing. The first thing they looked at was acidity. Using a special pH-sensitive dye that glows differently depending on the acid level, they could literally see the difference. The plaque that had been treated with arginine showed significantly higher pH levels, meaning it was much less acidic, after being exposed to sugar. The arginine-treated plaque was simply better protected from the acid attack. It was more resilient, but the changes weren't just chemical. They were structural too. Think of plaque not just as a blob of bacteria, but as a complex city with a scaffolding made of carbohydrates or sugars. These sugars, like fucose and galactose, help create what researchers call acidic pockets, which trap acid against the tooth. The team found that the arginine-treated plaque had less of the fucose-based carbohydrates, potentially making it less capable of trapping acid and causing damage. They also saw a fascinating reorganization. The galactose-containing structures moved away from the base of the plaque, the part touching the tooth, and shifted toward the top. This rearrangement could be crucial, as it might limit how much acid builds up right next to the tooth enamel, where it can do the most harm. Of course, the most important question is, did it change the bacteria? Using DNA sequencing, the researchers took a census of the microbial community. Both the Arginine and placebo-treated plaques were mostly dominated by common oral bacteria like Streptococcus and Veillonella. However, there was a key difference. Arginine treatment led to a significant decrease in a specific group of Streptococci that are known to be good acid producers, but not very good at neutralizing it. At the same time, it slightly increased the population of other Streptococci that are better at using Arginine to produce those helpful alkaline compounds. It's a subtle but powerful shift. Arginine wasn't wiping out all the bacteria. Instead, it was changing the team dynamics. It was tilting the balance of power in favor of a healthier, less aggressive microbial community. So let's put it all together. The study showed that in a real human mouth, Arginine made dental plaque less harmful in 3 key ways. First, it directly lowered acidity after a sugar attack. Second, it changed the physical structure of the plaque, making it less able to trap acid against the teeth. And third, it shifted the balance of bacteria, favoring a community that's less likely to cause cavities. This is huge. Tooth decay is one of the most common health problems in the world, affecting people of all ages. This research points to a new strategy that doesn't just fight plaque, but actually transforms it. The researchers suggest that adding Arginine to everyday products like toothpaste, mouthwash, or even chewing gum could be a powerful way to help protect people who are at high risk for cavities. And one of the best parts, Arginine is a completely natural amino acid. Our bodies make it, we get it from our diet, and it's considered very safe. This means it could be a fantastic option for everyone, even young children. This research offers a glimpse into a future where we work with our oral microbiome, not just against it. By providing the right fuel, like Arginine, we can empower the good bacteria in our mouths to protect us naturally. It's a smarter, more balanced approach to dental health. What do you think about this? Could Arginine be the future of cavity prevention? Let me know your thoughts in the comments below. Thanks so much for watching, and if you found this interesting, don't forget to like this video and subscribe for more deep dives into the science of our health. See you next time.